Thank you, Dr. Herrera, and for doing all the organization with your wife, Elba. We really appreciate it. It's been a wonderful meeting so far, and we hope to continue that. I've been told that I need no introduction, so I'm just going to begin and let you know about some of the things that we have run into in our many years of looking at clinical specimens, but along the way, sometimes we run into things that we are unfamiliar with. And it's not, it's you. I had set it up. Okay, but it's.
You have to use the keyboard.
Okay, got it. All right, thank you. So I challenge you to identify this unidentified creature, at least one of them in the bottom right-hand corner. Anybody know what it is? Especially the one with the funny hairdo.
David.
David in a different life. Yes. We've been to Madagascar many times. We love lemurs, and this is one of our friends. So, as you know, in transmission electron microscopy, we mainly use two techniques. Now, there are many techniques, but in diagnostic pathology, we do use negative staining. Our laboratory looks at a lot of virology samples, and we also use thin sectioning. So I will organize the talk in starting with negative staining. No, wrong way. And under each different category of method, I will organize it under viruses, bacteria, and higher organisms. With that said, with each different category of critters, I will give you a rule, an algorithm for looking for these things, and then I'll give you some examples of each one. So that's where we're going this morning, the outline. First, negative staining. The first question you ask is, how big is it? What is its size? If it's really, really, really small, it might be a virus. It might be something else, but it might be a virus. And so you have to know the size of viruses. Most come about 20 to 400 nanometers. There is one kind of a virus, a virus. You don't need to know that because most of them aren't pathogenic. That is on the order of 1,000 nanometers, and it was discovered within the last decade. So if it's small, it could be a virus, it could be some kind of cell component, and it's up to you to decide whether it's something to worry about. Shape is important. The questions you should ask are, is it spherical? Is it filamentous? Is it pleomorphic? Is it naked? Or does it have an envelope? I will explain those two in a moment. If it's enveloped, what does it look like on the outside? Does it have spikes? And what is its nucleocapsid on the inside? Nucleocapsid is that bag that holds the DNA or the RNA. And something about the uniformity of size. If they're fairly uniform, then they could be viruses. So here are examples for virus identification. Shape. Naked viruses are like a baseball. They have this hard outer coat that usually is roundish. They're spherical. Most of them are icosahedral. The large ones are on the order of 3/4 the length of this bar, or around 75 to 80, 90 nanometers. Like a baseball, they pretty much stay roundish in the stain as the stain is drying. On the right, we have the larger ones. These are about 3/4 of this bar. By the way, you don't need to remember the names of any of the viruses that I will mention. So rotavirus is not significant, except that it got its name because the structures on the outside radiate out from the center like the spokes of a wheel. Adenovirus is also icosahedral, and depending on how it lands on the grid, it may look to be hexagonal. And if you follow my pointer, you will see that it has flat triangular facets. Here's one facet. And here's another facet. So these are easily identified. On the other hand, the small round viruses, some are called small round viruses because when you look at them, they're just fuzzy cotton balls so that you cannot really identify them. I know this is a picornavirus because I took the picture. By the way, two things. If I don't have a reference, they're my pictures, pardon me, and they don't need a reference. If I stole it from someone else, there is a reference that you can photograph. On the left here, we have a small round virus that could be called a small round structured virus because some of the particles, depending on how they land, have structure on the outside that is recognizable. And you see that this has cup-shaped structures, and that's how it gets its name, kalisi, from the Greek word chalice. Another thing is the shape. Envelope viruses have this membrane around the outside that is pleomorphic. It's soft, not like a baseball, but more like a beach ball. And you leave the beach ball out on the sun for a week, and it takes all kinds of different shapes. This particular virus, this is flu, in some cases may look like a kidney, it may look like a peanut, and it may look like this big animal cracker down here at the bottom of the screen. Here. So envelope viruses are usually pleomorphic because they have this soft, pliable membrane on the outside. In addition, all of the envelope viruses have some structure on the outside. Sometimes it's easily seen by electron microscopy, and we call them spikes in the negative stain. These are the hemagglutinin, and the spikes are the things that attach the virus to the cell. And inside the virion means the whole virus, the inside, the nucleocapsid, the outside, the envelope. In envelope virus, we need to look at the nucleocapsid shape if it's visible. Here it is not. Here it is. So here's another example of an envelope virus. But inside this envelope virus, the stain has gone in. The envelope is this white area that I'm pointing to here. It's more visible down here at this arrow. This is the envelope that is also broken, and the stain has penetrated the virion. Inside the virion, the nucleocapsid shape is round or spherical, and it's 100 nanometers in diameter. This is herpes virus. So if I see a particle whose nucleocapsid is about 100 nanometers, it's probably in the herpes virus family. Now there's several herpes viruses in that family, so I couldn't look at it like this and tell you that it's herpes simplex or cytomegalovirus or EBV. But the nucleocapsid shape is important. The whole virus itself is called the virion. On the right-hand side, we have a nucleocapsid that is, again, proteins holding together the nucleic acid, which may be RNA or DNA, but not both in a specific kind of virus. And this is like a slinky or a coiled phone cord. So it is coiled up inside the whole virus, and you can just begin to see a few right here. This particle also has spikes around the outside. There are many things that can look like viruses. I call them masqueraders: cell components, proteins, lipids, vesicles, and membrane debris. So you have to sort out, if you're looking at a negative stain, what really is virus and what might be some garbage that is in your preparation, particularly if it's a stool sample. So the masqueraders, here's an example of one. All of this stuff over here on the right-hand side at the bottom is junk, cell debris. Now, how do I know that? there's one particle here that really looks like a virus. How do we know that? You, not knowing any more about virology, if you've never seen a virus before, could tell me that this set of particles is junk as opposed to virus. How could you do that? Look at the magbar. This magbar is the same size as this magbar. What does that tell you? That these two pictures are taken at the same magnification, right? So I've already told you that the herpes nucleocapsid is about 100 nanometers in diameter. Well, look at the thing inside of this particle that looks like it could be a herpes virus. My eye was drawn to this pile of junk over here on the right-hand side, but when I looked at the size, it's too small to be a herpes virus. So that's why size really matters in the case of virology. Here's another case of some junk. What about this? Would you say that there are virus particles here? What did I say about uniformity of size? Well, look at all of this junk over here. This is just cell debris. However, my eye was caught by these two sort of roundish particles here. And when I looked way over here on another part of the grid, I found clumps that were all uniform in size. And indeed, it being from urine was a polyomavirus. So moving up the food chain, we have an algorithm, rules for identifying bacteria. Again, size matters, shape matters. They can be spherical, they can be rod-shaped, they can be spiral or helical, and they can be sort of comma-shaped. Those are the four different shapes. Look at how they're replicating. Are they out there by themselves? Are they singles? Do they grow in chains like Streptococcus? Do they grow in clusters like grapes or Staphylococcus? Are they rods? What are the projections? Do they have pili or flagella? On this one, I have drawn a long tail representing its flagellum. So size, shape, how it replicates, and the projections. Here is an example of a bacterium seen from stool, and you see that it quite nicely demonstrates the pili, these little short, many-armed things. And here is a flagellum. So there's no question that this is a bacterium that we saw in a stool sample. Moving into the next technique, thin sectioning. Here are algorithms for looking at organisms by thin sectioning. Size, as I've said, is important. Is it bigger than a virus? Could it be something else, but still important? Maybe it's shown up in your tissue and something that should not be there. Shape, replication. How does it replicate? Where is it? Thin section lets us know where in the cell it is, or maybe it's extracellular. Something about its organelles or appendages can tell you whether it's a lower organism or a higher organism. The location in the cell, as I have said, is important. Is it in the nucleus? Is it in the cytoplasm? Or is it outside the cell? Other things that you can pay attention to in thin sectioning that you can't see by negative staining include, is it a prokaryote? That is to say, does it have a nucleus? Is it a eukaryote? Does it have a nucleus bound by a double membrane? Does it have a cell wall? And if so, how thick is that cell wall? How does it replicate? And does it have complex internal organelles? So here, dividing it up again, is viruses. I will tell you how to identify them. We've talked about size and shape. The naked or envelope, I've mentioned already. If it is enveloped, with which membrane in the cell is it associated? That's a clue as to which kind of virus it is. Where in the cell is it? How uniform is it? And something about the sample source. Some virus is home to certain systems, like you will never see rotavirus in the brain. So if you're looking at brain, you don't have to sort through your brain to determine that it could be a rotavirus. You can just cross off some of the names of viruses because some viruses aren't found in some systems. Here now are examples of those characteristics I just told you about. Of the naked viruses, size and shape are important. DNA viruses are usually assembled in the nucleus. Here's a cell. On the right, we have a nucleus, and this is a paracrystalline array, seen here at higher magnification. The viruses are in the nucleus, so that it has to be a DNA virus. Remember, viruses have one or the other, but not both kinds of nucleic acid. Now, it is possible. The caveat here, when I say usually, there is one caveat, and that's pox virus is a DNA virus, but it's made in the cytoplasm. So you don't have to remember that, but just remember that one can be out there. However, if the adenovirus here has made the cell so sick that the nuclear membrane is disintegrating, these viruses can leak out there into the cytoplasm. So the first thing you should do if you see a virus factory, like this RNA factory in the cytoplasm, is to look in the nucleus and see if they are also there. If you see viruses in the cytoplasm and not in the nucleus, it's got to be an RNA virus. So here are examples of envelope viruses. The previous ones were naked viruses. On the left, again, we have a DNA virus. Its nucleocapsid is going to be constructed in the nucleus. And it's going to get its envelope by budding through the nuclear membrane, seen here at the bottom. Here's one that's almost budded from the nuclear membrane into the cytoplasm. Here's a nucleocapsid in the nucleus, and here's a complete virion that's gotten itself almost into the cytoplasm. Now, herpes virus is sneaky. It can leak through the nuclear pores and get into the cytoplasm as a nucleocapsid unenveloped. But then it will bud into vesicles in the cytoplasm, or it may bud into through the cytoplasmic membrane into the extracellular space. On the right-hand side, we have an example of an RNA virus. It's out here, and you may not recognize it as a virus because this is an example of the measles virus that I just showed you by negative staining. It has the wormy-looking structures as the nucleocapsid, like a telephone cord. And here's the virus factory. And as these things get mature, they move toward the cell membrane, and it said, "Oh, cell, you're working for me now. You're going to put some of my proteins in your cell membrane, and I'm going to line up with those proteins and bud through the membrane." And so on the right-hand side, we have a higher magnification of the budding of virions into the extracellular space. As I said, some of the nucleocapsids are spherical. This is again herpes virus. Remember, these are about 100 nanometers in diameter. And these are helical. The helical ones, when you cut through a cell that's infected, look like you sliced through a can of worms. Some are in the plane and some are out of the plane. And then, of course, there are masqueraders by thin sectioning. And so you have to be aware that that's the fact. They're too numerous to mention. The most famous one, I think, is probably clathrin-coated vesicles. And you will see that in a later talk by my colleagues today. Other things include secretory vesicles. Dr. Wartchow mentioned them yesterday. He mentioned glycogen. He also mentioned nucleopores. Ribosomes are little dots, but they're not round, but they can confuse you, and microtubules cut in cross sections. Moving on up the food chain by thin section, we have bacteria next. Now, bacteria are bigger They're not as big as protozoa or algae, but they have this dichotomy that is one of the major differentiating factors by light microscopy of bacteria. Mr. Graham decided he could stain bacteria with crystal violet and they would turn purple. That's fine. But he also figured out that if you used alcohol to destain them, some of the bacteria would have their purple stain leached out, and they could be restained by a counter stain, safranin. So we have gram-positive bacteria that retain the stain, gram-negative bacteria that do not retain the crystal violet after alcohol and are red. Now, why do I tell you that? Because when you look at them by EM, you can tell whether the bacteria are gram-negative or gram-positive without the stain. So it has to do with the composition of the cell wall, the peptidoglycan layer here. Gram-positive bacteria have a thicker cell wall. Gram-negative bacteria have a different composition, and it's thinner. Here are examples, real examples of bacteria. On the left are gram-positive bacteria. We have a rod. You see this dark area around the outside is the cell wall, and it's fairly thick. There's a thick cell wall around this coccus. These are round bacteria or spherical bacteria that are reproducing by fission. So you have almost two here. You will see that it doesn't have a nucleus. These things in the middle are lipids. And on the right-hand side, we have rods. And look at the cell wall around the outside of this one. You see how that it's wavy and thinner, like I've drawn this red cell wall around the gram-negative bacteria. So if I saw this in a say a lung sample, I would know immediately that I have a rod. It's a bacterium. It doesn't have a nucleus. And I would be able to tell you that it's gram-negative. Here are other bacterial examples. This one is growing like grape-like clusters. It's pseudospherical or coccoid. This is a rod. It's gram-negative. This is a rod as well. But in addition, the one on the right has a capsule around the outside. And the capsule helps the bacteria evade the immune response. These are funny bacteria. They are bacteria, but they don't have cell walls. They are mycoplasma, and we'll have more to say about them in another talk later on. But you can see that these are stuck to the outside. They've attached themselves to this cell, but they don't have cell walls. Because they don't have cell walls, they can take a bunch of different shapes. Moving on up the food chain, we have higher organisms, and they are complicated. And I think the bottom line for higher organisms is look on the inside, and if they have a bunch of organelles that you don't recognize, you know it's something that shouldn't be in your liver, kidney, heart, brain sample. So the major characteristics for identification are, does it have a nucleus? What's the wall thickness? These guys are going to be larger, on the order of 10 to 100 microns, not nanometers. What are their shapes? How does it replicate? And especially look at the complex internal compartmentalization of the organelles. Here are examples of higher organisms. Yeasts are unicellular fungi. It reproduces by budding, as you can see here. And where is it? In the nucleus? Probably not, but in the cytoplasm or extracellular. And it does have a thick cell wall. Now, I took this picture in graduate school. This is in my dissertation. So that tells you that it is an ancient picture, but it's still relevant today. These are the yeast cells. And you can tell that this is the pseudomycelium and a spore that this yeast can make. And you can see how thick that cell wall is. Microsporidia are also unicellular fungi. They form spores, and inside the spore is something called a polar tube. And it coils its polar tube inside. It squeezes itself and coils it up. But if you cut this in cross section, you are cutting little saucers that are representative of the coils of the polar tube. Different ones of these have a longer or shorter polar tube. And so if you have 9 to 11, 12 of these little saucers on both sides, then it's one kind of organism. If you have 5 to 6, it's a different kind of organism. And I'm not going into which kind is which, but I'll show you an example in a moment where that was important. And these things replicate because when the spores germinate, they poke this polar tube out of the apical end, and the DNA, John Hicks, here's your snake, has swallowed a rat. We had a discussion this morning, and you can ask Dr. Hicks about that. The DNA from the spore is extruded through the polar tube that has attached itself to a host cell. The spore body doesn't go into the cell, but the DNA is squirted in there like a hypodermic. This is replicating in gut tissue. You see it's inside a vacuole. And here's an example of where the polar tube, when it was cut, is in 2 rows. You see here are 4, and here are 3 of those little saucers. So again, the numbers of coils of polar tubes are important in the identification. Now, we had a patient, an immunosuppressed patient, a transplant patient, where we found 2 different kinds of microsporidia. And we knew that because, as you can see, one has a lot of these little saucers, and the other one has only 6. I think there's 5 on this side and 6 on that side. So that let me know, and let the physician know, that there are 2 different kinds of microsporidia in this patient. Moving on up the food chain to protozoa, they are these big things. They do have nuclei. They're about 10 to 52, I'm missing the micron here, it was there, micrometers up to about a millimeter. They reproduce asexually, and they have many shapes. Now, I can't possibly go into all of the different protozoa, but the business end, the apical end down here, has a lot of organelles. So if you see something that's sort of banana-shaped, go to the pointy end and look and see if you see some things. I'll name them, but you don't have to remember. Micronemes, rhoptries, conoids, and these things are used for attachment and extrusion into the host cell. Here's an example of a toxoplasma, a protozoan seen in brain. It was taken by my colleague, Ted Wills. Some of you may know the name. Ted and I gave many tag team presentations at UltraPath. And this was a lovely organism, that picture that he took and was in some of our talks. Here are other examples of protozoal identification. You don't have to remember the names of any of these, but the idea is just that you look at it, it's big, it has a cell wall, and it's got lots of organelles inside, including a nucleus. And the final organism I'll discuss is algae. The size varies from really tiny, 200 nanometers up to 2 microns for the little guys. And kelp, seaweed, is up to 60 meters. Well, of course, that's not pathogenic, but the algae, the point is algae have a big range of organisms in that category. The things that are important here are that algae usually have starch granules and chloroplasts. Now, never mind the busyness of this cell. This is a diagram I stole of a plant cell, but the things that are important to notice in algae are that some of them have starch grains and or chloroplasts. They also have some crystals. So if you see needle-like crystals or granular crystals, it might be an alga. Again, don't bother with names of anything. Just look at the complexity inside. This is a chloroplast. It looks similar to a mitochondrion, except that the lamellae go up and down long ways instead of crossways like in a mitochondrion. There is one pathogenic alga, Prototheca, and it causes skin ulcers, septicemia, meningitis. So you will see it in tissue. It's usually found in sewage and soil. It's unicellular. It does not have chloroplasts. So if you see this thing, you would have to look for a starch granule. You would also notice that the complexity inside indicates that it's some kind of a higher organism. So you might want to ask the guy if he's been swimming in a pool somewhere or swimming in a lake. So to summarize, we've gone through negative staining and thin sectioning techniques. We've said that size and shape matter, how the thing replicates, and the things that thin sectioning can tell you here in orange that negative staining cannot are, is it a prokaryote or eukaryote? And the appendages on the outside, some you can see by negative staining, others, especially the complex organelles on the inside, you can see very well by thin sectioning. And with that, I will close and ask you if you know the difference between organisms here and which ones you might find in an ordinary household. And which ones are living and which ones might not be. Okay.